All right, well, thanks everyone for joining today to hear me talk. Today, I'm going to be talking about these RNA origami structures that I worked on during my postdoctoral fellowship in the Anderson lab. And I was able to characterize a lot of them with cryo-EM at a good time to submit them as targets for the CASP-15 experiment. So today I'll be discussing these RNA origami structures in the context of structure prediction. And so today I'll go over sort of the RNA origami design process, how we make our designs, how we use structure prediction tools during the design process, a little bit about RNA origami designs as a structure prediction target, and then I'll go over some of the differences between our designed models, our experimentally determined models, and then the predictions from the CASP experiment. But just to start, I'll discuss a bit the relationship between this folding problem that's the focus of the CASP experiments and this inverse folding problem, which is the focus of structural design. So with the folding problem, you're given this sequence and you ask the question, how will this sequence fold in 3D? And I think most of the CASP predictors would do a fairly good job at predicting this sequence to fold into this one 3D shape like a hairpin. And inversely, for structure design, we ask the question, what sequence will be able to fold into this shape? And for our purposes, we disregard individual bases and we just look at the overall structure. And so we can get this structural diagram, this dot bracket notation showing the base pairing pattern that'll give this shape. And then we design a sequence that'll fold into this shape. And with this simple sequence, there's many, many solutions that we could get to fold this shape. But what if we move on to a more complex sequence? I think maybe this one is a bit less intuitive of what it's going to fold into. And if we submit it as a target in CASP, we would probably get many different responses because there are many different possible base pairing configurations for this sequence. But maybe some of them would look like what we think the true structure is. And similarly, when we ask how do we design a sequence that'll fold into this structure, it's more complicated with these large designs. We have more complicated dot bracket notation, and it's more difficult for us to design a single sequence that'll fold into this structure and not misfold into alternate structures or get trapped in some topological trap. And so the question is, how do we simplify both the structure prediction and the structure design for these larger structures? And I think one way to go about that is to consider some prior knowledge about RNA. And one of the fundamental things is that RNA folding is co-transcriptional, which we can observe if we look at these beautiful Miller spreads of ribosomal RNA production. We can see as the ribosomal DNA is transcribed, we're already forming these dense globs of ribosomal RNA. And as the polymerase falls off, the ribosomal RNA is already mostly folded, and there's just a little bit of restructuring that has to happen. And so a consequence of this co-transcriptional folding is that RNA structures are actually modular. And I hope you can appreciate from how this rainbow coloring how individual helices are folded locally. So we have very similar colors on local helices. So most of these helices are formed with sequences that are close in the primary sequence. And we have relatively few long-range helical segments. One exception to this is this 5' 3' end down here at the bottom. And we try and do something similar when we design our RNA origami. We have these modular helical elements that fold locally, with some exceptions where we have to route complicated strand paths, then we get these more distally folded helices. And so I think when it comes to these longer, more complicated structures, instead of considering just what the minimum free energy structure of this sequence would be if it were to spontaneously collapse from being unstructured to structured, as we get to more complicated sequences that we want to predict or that we want to design, it'll be important to consider the co-transcriptional folding pathway. And this is what we try and do when we design the strand path for our RNA origami. So if we imagine this DNA gene and our T7 polymerase that we use to make the origami, we know the polymerase transcribes at about 30 to 40 nucleotides per second, and that the formation of helices and hairpins is a lot faster than this, on the order of microseconds. So pretty much as soon as it's transcribed, we have the formation of these local helices. And then the next thing that forms are these junctions, and they can take on the order of milliseconds to rearrange into how they prefer to stack. And a bit slower than that, we have these long distance tertiary pseudoknot or kissing loop interactions that form on the order of milliseconds to seconds. And then finally, we have this maturation of our fully folded RNA origami, which can then go on to assemble into higher order structures intermolecularly. And this architecture was published in 2014 by Cody Geary, Paul Rodemund, and Ebba Anderson. And they showed that using this architecture, we can get these extended lattices and really complex patterns and structures. And then as I was joining the lab in 2019, Cody was updating RNA origami to RNA origami 2.0, where they've now added a lot more different modules you can add on, protein binding motifs, aptamers, but also he really improved the software that we use to design these structures. So it's a lot more automated and a lot more high throughput. We have a piece of software, RNA build, that can take these blueprints and give a prediction of the structure that it'll form just using simple rigid body rotations. We have these substructure analyses that try and look at the co-transcriptional folding pathway. And we have our sequence design algorithm, which uses the Vienna RNA package to make sure that we're going to get the correct base pairing in our structure. And so now I'll just walk you through the RNA origami design cycle. So we start in 3D. We take RNA modules from structural databases, usually X-ray or NMR structures, but I think in the future it'll start to include more cryo-EM structures. And we orient these in 3D to align the backbone. And from this, we can create a 2D diagram of the base pairing patterns, and we can choose where we want to put the 5' and 3' ends in a way that will allow the local helix formation and co-transcriptional folding. And then finally, we design our sequence, transcribe it, and perform experiments to validate. And this forms the cycle of design, experiment, validate, tinker. Now, some of the motifs that we...
I have a quick question on the
sure.
slide. For your RNA modules, how high resolution or what type of detail do you need in those modules to be able to kind of use them? Like, do you need to have atomic level detail in those modules, or what is the desire, I guess? What is what would be the limitations if you had a lower resolution module to fit in?
I don't think we need atomic level resolution. In some cases, it might improve our confidence that our design is going to work if we do have that higher resolution. But as I'll get to later in the talk, in some cases, like high resolution structures might not be the solution structure either. So I think it's really context dependent. As long as we can fairly accurately place where the 5' and 3' ends are on a motif, then we could probably incorporate it into these designs. So one of the common motifs that we use in these designs are these so-called double crossovers. And what these do is essentially keep two adjacent helices parallel to each other. And the important parameter here in the design is that the distance between these two crossovers has to be an integer multiple of a full helical turn, or some multiple of 11 base pairs for A-form RNA. And so this next motif that we have is this 180-degree kissing loop that we borrow from the HIV dimerization initiation sequence that has this little double purine 6 base pair purine motif. And we basically use this to allow us to have a single stranded architecture rather than having multiple staples like you would with DNA origami. With this tertiary interaction, we can route everything on a single strand. And when we were choosing motifs, because this is such a well-studied structure, there are actually a lot of different structures in the database, most of them coaxially stacked in this 180-degree fashion, some of them bent. All of the NMR structures, interestingly, have these purines inside the helical axis, like so, whereas the crystal structures all have them bulged out and stacking with each other like that. And initially, we chose this X-ray structure to model in 3D for our designs. And the final important parameter that we control in all our designs is what we call the crossover seam. So if we have 3 or more helices, the number of bases between these 2 crossovers is going to dictate the amount of curvature between adjacent helices. So if you have something like 0 or 11 bases, you're going to have very slight curvature, whereas if you have something close to half a helical turn, you're going to have more of an acute angle between these helices. And so if we introduce a consistent seam length between these crossovers, we can get presumably a consistent curvature. And with 3 base pairs, we predict we'll get about 122 degrees of curvature. And if we repeat this 6 times, we should be able to form something like a nice hexagon. And so this is what we designed. And we created this base pairing blueprint for this design. I'll just walk you through some of the choices we made. So we chose to put the 5' and 3' ends here on this helix. You'll notice it's the only helix that doesn't have a kissing loop. And initially we'll transcribe this single stranded region, followed by this CLASP hairpin, which forms the first kissing loop, and the second hairpin, which now should coaxially stack with this other helix. And then we transcribe all these little hairpins that have these 3 nucleotide single-stranded spacers for now. So maybe these ones are less stably coaxially stacked, if at all. And then we get transcribing these internal kissing loops, and we can have another nice stacked helix up here. And then we continue completing these 3 nucleotide seams, getting all the internal kissing loops, then completing the 5' end, all these internal kissing loops, whoops, sorry, until eventually we transcribe this final half of this, this CLASP helix and go back and finish these 4-way junctions and finish molecule. Do you have a question?
Oh yeah, can I? Let's just, I try to imagine like folding of this. I mean, I know this is called transcriptional, but how easy is to fold a structure like that? Is this like super like stable? And do you have any measurements of like, how many of your, transcribed mRNAs that they fold into this shape? Like this is, I don't know, 10% that you will get there in some way or another, but it's so difficult to actually fold them correctly, or it's just so stable that pretty much like all of transcribed mRNAs will fold into something like that?
We don't have a great estimate of the yield of this particular one. So usually we transcribe and then we purify with size exclusion. And so you can see huge aggregates with that. You can't see minor defects really. And then our next step of characterization is with cryo-EM. And there we're discarding a lot of particles. Whether it's because they're misfolded or not, it's hard to say conclusively. But I would say for the most part, these fold pretty well, especially these shorter ones, around 700 nucleotides. Once we get over 1,000 into the 1,300, 1,500 nucleotide range, then we start to have like 20% that are clearly misfolded. So from this blueprint, we can generate these structural restraints. So this dot bracket notation showing the helices and pseudoknots, and then this sequence restraint. So we get a good transcription initiation. We have these nice UUCG loops and the motifs for the kissing loops. This gets fed into our sequence design program called Revolver, which will initialize a random sequence that follows these constraints. And then it goes through several stages of secondary structure prediction using the Vienna RNA fold software. So it'll start by mutating regions that are predicted to misfold that have GC base pairs. It'll iterate through this a few times. Then in the next stage, it'll look at anything that is predicted to be misfolded, and then either mutate those misfolded regions or alternatively do this neutral drift and mutate a region that is well folded, but just to sort of shift the energy landscape and find a solution to the whole problem. After it's satisfied, the minimum free energy structure is the same as our structural restraint. It goes on and does some GC richness reduction and pattern reduction, gets rid of any palindromes, repetitive sequences, restriction enzyme sites that we might not want in our DNA. And then in the next stage, it gets to mutating the kissing loop sequences so that they're orthogonal and we're not going to have any crosstalk between the different kissing loops. And finally, it tunes the energy of the kissing loops so that they're not all completely U or completely GC. And we usually run this 50 or 100 times, and we sort of rank the outputs we get by their predicted ensemble diversity and pick the sequence that has the lowest predicted ensemble diversity. And here is one of the sequences we submitted as a target in the CASP contest. And if you take this sequence and put it into almost any secondary structure prediction software, you get something that basically, predicts our structural restraint that we designed the sequence to fold into. So as a structure prediction target, it's kind of unique in that it has only one really satisfactory minimum free energy structure. So all the base pairing should be correctly predicted. And the challenge will come in correctly stacking across these four-way junctions and recognizing these kissing loop interactions. And so now I'll do a breakdown of the results from the CASP contest. So 3 out of the 23 groups that predicted for this sequence correctly predicted all 5 kissing loops, with an honorable mention to the Gene Silico lab, who got 4 out of the 5. And they seem to have stacked across these four-way junctions differently than what we observe. And as a result, I think they missed this final pseudo-knot interaction. The Chen group was very close to our design model, so what we used to generate our structure restraints. And the Aichemy2 and RNAPolis groups were very close to one of our experimental models. So here we can see the Chen model overlaid onto our model from RNA build, and you can see that the Chen group very faithfully listened to our RNA origami design principles and predicted this 122-degree crossover seam angle and got perfectly coaxially stacked helices. And unfortunately, things are never quite as you expect them to go when you do experiments. We took this 6-helix bundle to the cryo-electron microscope, and this is what we see in our ice. We have some things that look like they could be tubes sitting on their ends and some that could be tubes laying down. When we pick these particles and do these 2D averages, we can see, some nice tube structures laying down, but then we see some weird angled protrusions that we wouldn't expect from our design. And from this really informative 2D class looking through the helical axes, we can clearly see there's 5 helices and like a smear over top. And when we reconstruct in 3D, we get something that looks like this, where we have the clasp helix sort of at an angle to the rest of the bundle, which when we thought about in retrospect, makes sense because we no longer have this double crossover tying this helix down parallel at both ends. It makes sense that it can have this, this angle. And so satisfied with the structure, I went on to try and add some protein binding motifs on the ends of these helices. And so I purified the RNA and purified some protein. And while I was purifying the protein, the RNA sat on the bench for a while. And then we incubated them. I ran a gel to see if it was shifting, and the RNA sat on the bench for a bit longer. And eventually we tried to purify the RNA protein complex. And by the time it had gotten to the plunge freezing stage, it had been at room temperature for quite some time. And when we reconstructed the structure, although there's no protein, we did notice that this clasp helix is at a pretty different angle than it was in our initial design. And of course, there's two possible explanations. There's the amount of time it spent at room temperature. Maybe it matured into a more parallel structure, or maybe it was the effect of the protein and the protein binding motifs that we added. So we went back to our original design with no protein binding motifs, and we just let it sit at room temperature for about half the time that this construct did. And from this data set, we were able to reconstruct two different conformations. One that looks like the previous structure, the first structure that we saw, and one that more closely resembles this, this structure with the protein binding motifs. It doesn't look like this kissing loop is quite as coaxially stacked as it could be, so maybe this is not quite fully matured. Then we thought, since this is such a large conformational change, maybe we can observe this with small angle X-ray scattering. So I purified some more fresh 6-helix bundle, handed it to the SACS experts at iNano, Jan and his student Helena. And Helena measured the RNA in 15-minute data collection windows overnight. And what we saw was a decrease in the high angle scattering over time. And when we look at the predicted scattering from our PDB models of the early and late structures, we see that they overlap nicely with our early data frames and our late data frames. And then if we look at this particular scattering angle where we see a large amount of change, we see it starts as this like metastable state, transitions, and then plateaus again. We've since then sent this sample to other collaborators at Berkeley who have also been able to follow this transition with cryo-EM. So we were fairly confident that what we were seeing was a structural transformation, and we submit this target to CASP as a structure that we have from an early state, and then 8 hours later, it's in an alternate conformation, and we asked the predictors to try and predict both states. And we saw one prediction of the early state from the Aichemy RNA2 group, where we don't have this kissing loop even formed yet. We think this is really early. Probably this kissing loop forms before the structure is finished being transcribed. But we saw no other attempts at anything like an early structure. But we did see two attempts that were very close to our mature structure, which we were very enthusiastic about. So here you can see our cryo-EM reconstruction and the Aichemy prediction, and also the RNAPolis prediction. And they're pretty close. But if you look at some of the details, for example, this helix 5 here we have is quite curved, whereas the prediction has it pretty linear. Both predictions have it pretty linear. Similarly, if we go and look at helix 3, it's very banana shaped, whereas the predictions don't quite capture this. Helix 4 here has a pretty substantial kink in it that we see. And again, we just have pretty linear helices in the predictions. And finally, helix 1 is also curved, not in the middle, but at this end near the kissing loop, or near the crossover, sorry. And the predictions are of pretty linear. And we think we have an explanation for this, but yeah, we were really happy that these predictions were able to get closer than our design to the experimental structure, as it really holds promise for using these predictions in 3D to help design RNA origami in the future. But we think we potentially have an explanation for why the predictions didn't capture the curvature in the helices, which I'll go over with you now. So from one of our higher resolution structures with this 5-helix tile, especially on the central 3 helices, we were able to get down to about 3.5 angstrom resolution, which was enough for us to model the kissing loops with slightly more confidence. And what we saw was that these purines are actually on the inside of the helical axis, with these A2s stacked inside the major groove. and with these A1, A3 trans Watson-Crick base pairs forming, just like we see in the NMR structures. And importantly, if we look at the X-ray structure in our map, we clearly don't see any density for these bulged out adenines. And if we look at the distance between adenine 1 of the motif and then 3 bases after the motif, we have this distance of 30.7 angstrom. Whereas if we look at the same phosphate distance in our cryo-EM model, we see that it's closer to 27 angstrom. So essentially, in our design, we had thought this structure would take up one more base pair of sequence space. And because it's really important to have integer number of helical turns between the double crossovers, we think this is what was causing the twist defect. And we observed this twist defect in all of our structures. Unfortunately, we characterized a whole suite of structures before we got this high resolution structure that gave us this extra information and an idea of how to correct it. But we consistently see this twist across all of our structures with the original design, which is exaggerated in structures that have multiple of these kissing loops. And if we look down the helical axis, we can see not only is this severe seam twist present, but our inter-helical angles are also thrown off. Yeah, Marcin.
Can we go back to slides, I think? To the comparison between and here, yeah, do you think it's possible that the X-ray structure is actually like I don't know, in some kind of confirmation that actually if you go back and do cryo-EM for this fragment, that actually you will see that there are these two states, what you see in cryo-EM and what they could see in this RNA, that, you know, that maybe, you know, the other CASP line, you know, assembly, that's something to look for, like, you know, because sometimes the structure, they may have some this crystallographic contacts and things like that. Do you think that it could be some artifact or like some one of the states for this RNA?
So across all of the kissing loops we looked at in our RNA origami structure, there was really only one example of something that didn't clearly look like this sort of kissing loop.
Okay.
And we think that we could see half of these adenines bulged out and the others were inside. So it wasn't even fully like the crystal structure. But we think for the most part, at least in the context of our RNA origami, they're in this conformation. I don't know, maybe if you had this motif in isolation and it wasn't restrained on both sides by other sequence, maybe it's more prone to forming these bulged out. It's hard to say.
Yeah, that would be interesting. Yeah, no, Thank you.
Yeah, so just by going back and adding in an extra base pair before each of these kissing loops, we were able to create this twist corrected structure, which appears to be a bit overcorrected. Instead of twisting up, it twists down a little bit now. And our inter-helical angles were much closer to how we designed them. And so we think, because all of the CASP predictions of the 6-helix bundle used the crystal structure and had these bulged out adenines and this distance of about 30.7 angstrom, as I show on this slide, that could be why they predict perfectly straight helices and didn't capture the bend. It could be interesting to see what they predict if they use our cryo-EM model of the kissing loop to seed their predictions, to see if they predict the bend in the rest of the helices if they incorporate that. Yeah, so just to sort of wrap up now, discussing RNA origami as a structure prediction target and some of the challenges and simplifying factors. So I think a big challenge for the deep learning approaches is that as a completely synthetic sequence, you can't do sequence alignments. There's no evolution information you can use to infer which bases are important. They're also typically very long structures, over 300 nucleotides, and most of the structures solved for RNA are a lot shorter than that. Many pseudoknots, tertiary interactions also complicate things. But some of the simplifying factors, obviously the sequence has been optimized to reduce the ensemble diversity and to have a higher chance of having this one structure. There's usually no single-stranded or flexible stretches, so not a lot of large-scale dynamics are possible. And a lot of the motifs are from the PDB. So like the tetraloops and kissing loops you can pull from the PDB, but this is also a challenge. For example, the 6-helix bundle, as the kissing loops from the PDB are different than we observe with cryo-EM. Finally, we do get a lower resolution structural information from cryo-EM in general for RNA. So while we don't have details of individual base positions, we frequently get between 5 and 6 angstrom resolution, which is enough to fit helical elements and get an idea of the overall shape of the RNA. And also RNA is dynamic. So here we've shown the other 3 targets that we submit and this 3D variability analysis we did on them, which basically just tries to use principal component analysis on a full particle set before we've done any sorting in 3D. And it takes particle subsets along this trajectory of variability, and we can reconstruct different models from different particle sets and get an idea of the amount of movement. So going forward, I think incorporation of some of this dynamics from 3D variability analysis or perhaps other ways of validating dynamics could be important for structure prediction. Also, structure prediction and design considering co-transcriptional folding is going to be important for delving into longer, more complicated structures. And I guess just a personal goal, I think we need to solve more RNA structures to expand our design toolbox and also improve our structure prediction skills and what we understand about RNA folding. And then I would just like to acknowledge everyone in the Anderson lab, especially Eva, Cody, and Nestor, who I work really closely with on a lot of this, our collaborators who perform the SACS experiments, everyone at the MBION cryo-EM facility in Aarhus, funding agencies who supported this work, and then Houston Methodist, where I now am, and funded by the Cancer Prevention Research Institute of Texas. And I'm happy to take any other questions if there are.
All right, thank you very much. So everyone feel free to raise their hands or type questions in the chat and I can read them out. I'll just go ahead and get started and talk a little bit more about, I guess, HIV-DIS and more generally the motifs that you use. So it seems like from HIV-DIS, the NMR structure would have solved some of the problems. Did you see a similar trend in any of the other motifs? Or I don't know if any of the other motifs actually have both NMR and X-ray.
Most of the other motifs we use are like aptamers and things for which there are only crystal structures. We haven't gotten any of them to high enough resolution where we can see any differences. And we haven't noticed any. Usually we place them on the ends of helices too, so they're not going to have this effect of being in the middle of a double crossover where we see this difference in the spacing. Mm-hmm.
I think, yeah, there was before from Chaitanya. So I don't know if I pronounce your name. I think you raised your hand first. Can you ask your question? Sorry. Yeah, it's hard to. Yeah,
I was actually clapping, but I do have questions. I had a potentially dumb question. So you're taking motifs from the PDB and stitching them together, but these motifs would also have sequences associated with them, right? So why did you need to go through this pipeline where you're going to the dot bracket notation and then redesigning this sequence?
So I don't think it's a stupid question, first of all. It's a fine question. So some of the motifs have specific sequences that they need to have, like certain aptamers and things. But a lot of the structure is made up of just double-stranded RNA, which can be any sequence. And so in order to control how it folds, we need to redesign it so there's no overlap with other areas. And for example, the kissing loops, we want them to be orthogonal to other kissing loops. So we need to design the sequence so that it'll specifically base pair with its partner and not with other kissing loops. And we don't get this like mangled, weird kissing pattern. Does that answer your question?
Yeah, that does. And thanks for the great talk as well.
Thanks.
All right, Steve, next.
Sure, thanks. Thanks for the great talk. So I was wondering, this kissing loop motif that you showed that had like discrepancy between the X-ray and the cryo-EM. So how many instances of that specific sequence are there in the PDB? Or is there like, does that particular kissing loop show up many times in the PDB, or is it just from a single structure?
Yeah, there's a lot. I don't know the exact number, but it's there are structures of it bound with different ions and different drugs and things like this. And it's pretty consistent that across all the X-ray crystal structures, you have these bulged out purines, and all the NMR structures, you do not. There's more deviations between the different NMR structures in terms of how coaxially stacked the helices are. But,
but in terms of also, it's also, you also find the same kissing loop sequence within different types of RNA structures. Like,
yeah, so they're pretty conserved across different families of viruses. So I think there's a couple of variants of the kissing loop sequence in HIV. Is that what you're asking?
Yeah, well, I was just wondering, because I was wondering how much could there be this, how much does this discrepancy that you saw is a factor, or how much is it context dependent, right? So because like this idea of building RNAs or predicting RNA structures from motifs is highly dependent on this idea that these motifs are highly modular, right? Like you're putting them like Legos into different things. But then that becomes a big challenge if these Legos assume different conformations depending on where they're placed. So yeah, I was just wondering if you had any thoughts on that idea of how modular these 3D motifs are.
Yeah, we think the kissing loops are pretty consistent, and their structure isn't dependent on the kissing sequence. And I have a bit of data I can show to support that. So with this 5-helix tile, we made a design that had only different kissing loop sequences. You can see we still have this twist defect that we observed. So yeah, basically we think that the sequence isn't going to affect this kissing loop structure.
Sorry, I didn't mean the sequence of the kissing loop. I meant that the rest of the RNA, the surrounding stuff. Could that, if there's like if that helix where the kissing loop is embedded is let's say it's interacting with the neighbor helix in some way, is that interaction could that end up altering the structure of the kissing loop?
Yeah, I definitely think so. And especially having the structure being constrained on both sides by this sort of scaffold around it, that could also, introduce differences in the structure if it were, say, just free floating on its own.
Thanks. Thank you.
All right, John.
Hi, thanks for the talk. Really learned a lot. Several questions, I guess. On the folding intermediate, do you have any sense of what the free energy difference might be between that state and the final state?
It's not something that I've calculated or would feel confident calculating accurately myself. We think that the transformation is going to be driven by things like sharing of cations. We think that it's going to be driven by magnesium, and that it's going to be fairly entropically driven rather than through increasing the number of bonds. That's our idea, yeah. But we don't have any free energy calculations to support that.
Are there other RNAs where these sort of intermediates have been seen?
Yeah, I think this is probably the slowest maturation process that I've seen, but there are some that are on the scale of hours. And there are other examples recently with cryo-EM where you see trapped intermediates, usually from a heat annealing process more than co-transcriptional folding, at least the examples I'm thinking of.
I see. So I mean, this is sort of another vague question, but is these sort of intermediates don't seem to turn up very much with proteins, at least the proteins I'm familiar with. Do you think that RNA is more likely to have these kind of things? Is there any reason for that, or is that just too vague to address?
It's hard to say. Like, I think we're pretty lucky in even noticing this maturation. It's not often that you look at a sample, that's fresh, and then you look at it 8 hours later and get these two structures. So there could be a lot of things we're still not aware of that are doing similar things. I'm not sure if you would find this in proteins or not, but I think we were lucky to find it in this particular structure. And of course, it could be completely artificial and induced by the synthetic nature of the sequence, rather than something that would naturally arise.
Well, but that would suggest that somehow natural sequences are selected to avoid those kind of things, right?
Mm-hmm.
Which again is not sort of a trendy thing to say about proteins, but okay, thank you.
But I don't know how many predictors we have, but I really enjoyed this point on predicting structures, like design structures and structures like natural structures. So that would be, I don't know, if there is any way to test it. I don't know if there are any predictors that they have any, ideas or feedback, what was easier to predict, because I think, yeah, there are like these rules that that the natural structures they have to follow, and it may be more difficult to model, but actually if you see something that this is designed, that it could be much easier to be folded. But yeah, I don't know how to measure that, if there is any way to do some computational experiment and to really show this. Because another thing that I now recall, that when Andriy Lupas was talking about AlphaFold, it was interesting that I think his argument was that actually AlphaFold was really good at predicting design structures, because they were like kind of that they follow some kind of rules of nature to some extent, and AlphaFold was able to learn that. So without even deep alignments, they were able to predict some of the structures. This was like a few years ago or 2 years ago. So I wonder if this is if this is still something that people feel about AlphaFold. But yeah, I don't really know about RNA. Mm-hmm. But I don't know if there is anyone in the audience from the predictor side to comment. I'm not sure. There are so many names, but it would be interesting to also to see if they could comment at some point, if this is easier actually to without MSA and without this evolutionary information, that actually this is kind of easy to understand what kind of rules people were following designing structures. So maybe in new rounds of RNA puzzles, we can look more carefully into that. That will be interesting to see.
Well, I think the added layer on that you nicely in your talked about a lot is just the co-transcriptional, right? I think, whenever I predicted in RNA puzzles, I've never, considered co-transcriptional, even for stacking, it seemed very important in your designs to really consider that they were co-transcriptionally made, and that was the driving force on what stacked on what, and not kind of the normal energetics. And at least in any of my predictions, I've never considered co-transcriptionally, because so many of our RNA structures previously are just denatured and refolded, right? So it could be the case that even natural RNAs, maybe the difference isn't natural versus design, but there's also a difference co-transcriptional versus denatured and refolded. I don't think there's many of us that in our pipelines experimentally actually try to solve the co-transcriptional product.
Yeah, and I think we don't really have tools for RNA-PD structure predictions. I think kind of like in the field that they really take into account co-transcriptional folding, I think. kind of, that would be also yet another thing to test like computationally.
Question for Ewan. Have you guys tried denaturing and then refolding? Or on the other side, have you done designs where the stacking is less kind of decided by how you're actually making the RNA, right? You're not purposely designing those 3 base pairs to stop the stacking and everything, and has it kind of, do you get much poorer performance? How important is that stacking, co-transcriptional stacking consideration in your designs?
So for your first question, we've tried refolding a couple of them, and it seems to work pretty well for 2 helix tiles and even 3 helix tiles sometimes, but anything above that, we pretty much don't get a nice structure back out of it. And your other question about how much we consider this stacking across the junctions, actually, surprisingly, even with 0 base pair crossover seams, we get the stacking that we designed. So I don't know how important that 3 nucleotides of single stranded that I showed during my talk is for actually getting the right stacking alignment, because even without it, we still see the stacking that we've designed.
All right, Andre has a question.
Yeah, really nice talk, Ewan. Thank you so much. So I'm just curious, once you notice these weirdly looking particles in your eyes and you started thinking, what can be the reason for those? So you turn to Sachs, and Sachs beautifully confirmed what you thought. So do you think there are some other, so that's nice that Sachs was so useful in your case. So do you think some other experiments, like low fidelity experiments, could also help you in that, like cross-linking or something similar to that?
You mean some experiments where you put some labels on the structure to try and monitor the change?
Well, yeah, you don't see, them exactly, right, in your cryo-EM data, and you think, what would be other way, to confirm my hypothesis, right? So Sachs helped you, But let's say cross-linking, can you put some labels on them and think, and see, if this confirms what you think about your particles, your structure, or cross-linking is too short in your case for RNA, so I don't know.
We thought about some other approaches. For a while, we were considering doing some Fred experiments where we can put like fluorophores on the helical termini, and then we can see them get closer to each other. But the site-specific labeling on such a large RNA is kind of complicated and expensive. And so we gave the Sachs a go, and it worked. So we didn't give it too much thought of, how else to validate it.
Mm-hmm.
Yeah.
Thank you.
So can you maybe briefly, if there are no other questions, to explain what you're going to do? Because I believe you're starting your own group in Texas. So what are you going to apply some of your structures into like some applications?
Mm-hmm. Yeah, so I can give a brief spiel on that, I guess. So I think currently RNA therapeutics and mRNA therapeutics are like a really exploding field, and a lot of emphasis has been put on the specific sequence of RNA that you use as a therapeutic, but not so much effort has been put into the structure of these RNAs. There's some work going on about optimizing the secondary structures inside coding sequences to improve translational output and things like that. But what I'm interested in is designing structural elements for 5' and 3' UTRs that can control translational output in specific cellular contexts. So that's one of the main projects that I'll be working on going forward.